Our second class of biological organic molecule are lipids. Lipids are typically long hydrocarbon chains, so they are primarily hydrogen and carbon. They can be linear chains or they can be rings, but because they are primarily hydrogen and carbon, they are hydrophobic. So this is the characteristic of lipids. They will not mix with water. They are very nonpolar, very hydrophobic. They are used for energy storage primarily, but also some important structural components. Common fats and oils are called triglycerides. So cooking oil, for example, is a triglyceride. So we begin on the far left with a molecule called glycerol, and we are going to add to that 3 fatty acids. These fatty acids typically have very long hydrocarbon chains, and on one end there is an acid group, and so we will have the same dehydration synthesis reaction 3 times, and that adds these fatty acid tails to the glycerol. A lot we hear now about the difference between saturated and unsaturated fat. If you look at the top 2 fatty acids in this diagram, and notice there are no double bonds between the carbon atoms. And because there are no double bonds, those fatty acids are saturated with hydrogen. These molecules are a solid at room temperature because the fatty acid tails can pack together very closely in a very dense molecule. The fatty acid on the bottom has a double bond between the carbon. And so as a result of that double bond, it is missing a hydrogen. So that would be an unsaturated fatty acid. Whenever you place the double bonds in these carbon chains, it actually causes them to bend. That chain is no longer a straight line, and so these double bonds cause these fatty acids to bend. They can't pack as closely together, they're not as dense, and so consequently they are liquid at room temperature. The danger with saturated fats is they are also a solid at body temperature. So the difference between fat on a steak and vegetable oil, the vegetable oil has a lot of unsaturated fatty acids. Animal fat is typically saturated, so it is a solid. When we talk about lipids, we also need to mention emulsifiers, and these are some small molecules that will cause fat to mix with water. They're like detergents. This is the way that detergent works. It's a small molecule that has both a polar end and a non-polar end. So whenever we mix a fat with an emulsifier and water, what happens is the polar end of the emulsifier will mix with water, the non-polar end will mix with the fat. And so the fat still does not dissolve in water, but it's broken up into small droplets that can disperse in water. Another very important lipid that we will be discussing in the next 2 chapters is a phospholipid. This is the primary molecule that makes up the plasma membrane. There's a diagram here, and if you look up at the top, there is an area that we call the polar head. This polar head contains lots of oxygens. It even has some full charges, so it's very polar, very hydrophilic, is going to like to mix with water. And then attached to that are 2 long fatty acid tails, which are very nonpolar. So we call this an antipathic molecule. That means it has one polar end and one nonpolar end. So again, this is a very important molecule that we will see in chapter 3. Steroids are also lipids. In this diagram, each of these yellow rings represents a hydrocarbon ring. These rings are fused together. There are a couple of oxygen atoms on some side chains. However, the bulk of this molecule is a very hydrophobic nonpolar hydrocarbon chain. Cholesterol is an important structural component of animal cell membranes, which is a lipid, and then that is also the precursor for some important hormones in the body. So pictured here is both cholesterol, and then with some modification that can become testosterone. Our next class of molecules is proteins. Proteins are polymers of amino acids. Amino acids all have this same basic formula. They have an NH2 or an amine group on one end, and then a central carbon, and then a carboxylic acid, COOH, on the other end. On that central carbon, there is a variable R group. There are 20 different amino acids, and they vary because there are 20 different R groups, variable groups. So here are just a couple of examples. If you look at these formulas, you can see the NH2, the amine. On the opposite end, you can see the acid group. And then what's shown in blue is some of the different R groups. So the first one here would be the amino acid alanine. The second one would be valine. They all have the same backbone, the amine, the central carbon, and the acid, and they only vary in the R group. To make a protein, we're going to take these 20 amino acids with different R groups, and we're going to hook them together. Now, it's also important to remember that these R groups are variable. Some are charged, some are polar, some are nonpolar, and that is very important because the nature of that R group is going to determine what this amino acid wants to do in an aqueous solution inside water. If it is a nonpolar R group, it'll try to get away from water. If it is a charged or a polar R group, then it will mix with water. So to put amino acids together to form a protein, it's the same reaction that we've seen. It's a dehydration synthesis reaction. So on the left side of the diagram, we have two amino acids. We're going to take an OH from one, a hydrogen from another. We break those bonds to form a water molecule, and in doing so, we form a new bond between the two amino acids. So two amino acids would be hooked together. We would call that a dipeptide. If we continue this reaction, we make a polypeptide or a protein. Functions of protein are varied. They're structural components in the cell. They are hormones, which are the chemical messengers. Proteins are involved in movement, both intracellular movement and movement of entire organisms. They're involved in transportation. Our antibodies, which are involved in our immune response, are proteins. Enzymes that we will see in lesson 5 and 6 are proteins. So a lot of very varied functions. Also some very important components of the cell plasma membrane that we will see in chapters 3 and 4 involve proteins. Proteins have 3 or 4 different structures. The primary structure is simply the sequence of amino acids. So each of the little balls here represents an individual amino acid. It's that same dehydration reaction over and over, puts these together. So this primary structure is the amino acid sequence. This is determined by genes. But proteins do not exist as extended chains. Instead, they fold up. And first they fold into secondary structure. Secondary structure can be a helical structure, or it can be a structure called a pleated sheet, where the amino acid chain folds back on itself. So as the proteins are made, that amino acid chain will fold into one of the secondary structures, depending on the R groups and how they want to interact with water. And then that secondary structure will continue to fold, and it will fold into something called the tertiary structure, so much more complex folding, and in some cases, quaternary structure. Quaternary structure involves 2 separate polypeptide chains. Each of those folds into its own secondary and tertiary structure, and then 2 or more of those get together to form the actual functional protein. Shape is very important all throughout biology. Structure determines function. And so the way this protein folds up into this specific shape is going to determine how it acts and how it functions in the cell. Shape determines function. Our last class is a nucleic acid. Nucleic acids are the RNA and DNA These are polymers of nucleotides. A single nucleotide contains a phosphate group that's bonded to a 5-carbon sugar called a pentose, and then there are 1 of 4 nitrogen-containing bases. Adenine, thymine, guanine, or cytosine are the 4 bases that are found in DNA, and there's a 5th base called uracil, which is found in RNA. DNA stores our genetic information, so the chromosomes which store our genes in the nucleus are made up of DNA, and those genes are the instructions for making proteins. It determines the sequence of the amino acid. So the sequence of the gene determines the sequence of the amino acid. The sequence of the amino acid, remember, determines how it folds up, and its shape determines function. One last nucleotide that we will be discussing in subsequent lessons is a nucleotide called ATP, or Adenosine Triphosphate. It contains the base adenine, one of those 4 bases we mentioned a moment ago. It's bonded to the pentose sugar, and then there are phosphates attached to that. ATP contains 3 phosphates. However, this is how we store energy. The ATP can be broken down into ADP, adenosine diphosphate, and whenever we break that chemical bond, that releases energy. So whenever we have excess energy from cellular metabolism, we take the ADP and form ATP by forming that covalent bond between those last 2 phosphates, and then whenever we need energy, we can break that bond to release the energy.